So meiosis, just like mitosis, is preceded by chromosome duplication. So that means that before meiosis, just like before mitosis, we have to make a copy of all of our DNA. However, one of the things that makes meiosis very different is instead of just having one division process, there's two. So we divide one cell into two, and then each of those cells gets divided into two for a total of four daughter cells. The first division, which is called meiosis 1, so not meiosis I or meiosis L, that's basically a Roman numeral 1. So in meiosis 1, we start with the pairing of the homologous chromosomes. So the chromosome from mom and the chromosome from dad join together. Then there's a process called crossing over that occurs. In crossing over the homologous chromosomes, so for example, the chromosome from your mom and the chromosome from your dad, they exchange or they trade parts of their DNA. So meiosis 1, when it's all finished, it separates into two daughter cells, and each of those daughter cells has one set of chromosomes. In fact, each of those daughter cells would be, even though it says just one set of chromosomes, remember they're duplicated. So it's actually the same amount of DNA as a diploid cell. But since they're not separated yet, this is where it gets a little confusing. We could say that they're haploid. Meiosis 2 is similar to mitosis. We have to actually split apart the chromatids. And the result of meiosis 2 is that we have four haploid cells. So each of these cells will just have one set of DNA. So for the individual steps, it's going to get a little bit more confusing than mitosis. So with interphase, the cell looks the same. We can see the DNA is in the chromatin form, so it's very loose and thin, and it looks like spaghetti. But as we go into meiosis 1, we're going to see something that we've never seen before. When the chromosomes condense, the homologous pairs, so for example, chromosome number 7 from your mom and chromosome number 7 from your dad, they actually join together and connect. And they form a structure that looks like this, which is called a tetrad. Tetra is a prefix meaning four. And the tetrad is actually made of two duplicated homologous chromosomes for a total of four individual chromatid, two from your mom and two from your dad. And the points where they are connected, this is where crossing over is going to occur. Crossing over is where the two duplicated chromosomes can actually trade their parts. So at the end of meiosis, our DNA molecules are actually quite different from when we started. The DNA from your mom and the DNA from your dad actually gets mixed together. So when you make gametes to pass on to your babies, they don't actually get an exact copy of your dad's DNA or an exact copy of your mom's DNA. Instead, they get a blend or a mix of your mom and your dad's DNA. And that's because of crossing over, partially because of crossing over and some other things that we'll talk about. But after that, the steps are quite similar, but because this tetrad formed, one of our division stages has to actually separate the tetrad. So if we look here at prophase 1, so this is different from prophase. It's also different from prophase 2. So there's three types of prophase: prophase 1, and prophase 2. In prophase 1, which is the prophase of meiosis 1, we see the homologous chromosomes join together into the tetrad structure. Just like with prophase of mitosis, the nuclear envelope starts to break down and the spindle fibers are starting to form. When we get to metaphase 1, the tetrads will line up in the middle, as opposed to in mitosis when just the chromosomes lined up in the middle. And when we look at anaphase 1, we're actually pulling apart those tetrads. So when we pull them apart, we end up with molecules that actually have two chromatid. Then we'll take a look at meiosis 2 next.